Christine drove that, and really, Dr. Grace really was amazing and brought us in a network of coders, and that was an incredible event. We have a rain benefit concert coming up. There'll be not a lot of science, there'll be a lot of geekiness, but this is an incredible band that's going to be here. Come support us. There'll be lots of food. There'll be some drink. Ricardo is, believe it or not, a local artist. He does he's played around here. He has an extraordinary band he's bringing over from Hawaii. He has a studio there. He usually plays at the Triple Door up in Seattle, and he's coming over specifically to help out Rain. So please come out. It should be a really fun night. There'll be a lot of dancing. Bring kids. It should be great. So please come support Rain. We want to talk about our iGEM experiment fundraiser. We have a high school team, the International Genetically Engineered Machine. If you do not know about it, it is an incredible competition, about 350 teams from all over the world. And we go to Boston and compete with Pasteur Institute, Stanford, University of Washington, Seattle has a team, MIT has a team, and so it's all about synthetic biology, which is fundamentally what we do here. We teach skills in synthetic bio, and so this is great skills. If you don't know what synthetic bio is, ask anybody here with a Rain t-shirt. Ask any of our scientists here, we'll tell you about it. The team this year is working on a project for nitrogen fixation to enhance that. Basically, they're terraforming a planet, I like to say. So these are important concepts done on a very small scale. They can do huge things. And our kids are learning the skills and driving this to be competitive in the future world. So go to the site. It goes live tonight, Matt. Is that right? Yeah, it goes live tonight. And we need to raise some money to get teams to Boston. I have to also, the Vibe, the Veterans Incubator here for business entrepreneurship with the University of Washington is going to be here for a business competition this week and next week. So there's prizes, there's teams involved. It's super interesting, super competitive, and it's going to happen right here at Rain. So come on and support this. Finally, our next superhero, well, pentultimately, I guess, our next superhero is Josh Santarpia. So he's a returning superhero. He has a new position. He's left Sandia Labs, and he is going to University of Nebraska. And so he he basically heads up strategic bio. So he's a DOD or DOE employee. He talks about all kinds of pollution tracking, chemical threats. So he basically does threat detection, but a lot of in the context of environmental monitoring, whether it's pathogens or chemicals for pollution or to chemical warfare agents. So he builds all kinds of systems for doing that. Okay, let's get on to our superhero of science tonight. So I am really excited to present Cindy Lawley. So Cindy comes to us from San Diego. She came up specifically to come give a talk to you guys tonight. So Cindy, I think above all else, is a high school science teacher. And I'm just really excited about that. So in addition, she's a professional diver. She's a marine biologist. She has a huge long list. Anyway, in addition to that, she's gone back to school. She got her PhD and had a completely new career at Illumina, which is a big sequencing company. And so she is just extraordinary. I got some chance to talk to her tonight. I've talked to her on the phone several times. She has written a talk from scratch because that's what a science teacher can do. And so it's very exciting. I encourage you to talk with her afterwards. We are going to have questions and answers afterwards, but we'll probably, feel free to leave when the talk's done. But if you can stay, I promise you I'll be the last one here to leave. So if you want to stay and talk to people, okay? So, Cindy.
I cannot tell you how much I am excited to be here. And I thank David and Mary Lynn, who connected me with David, for helping me see the opportunity and be able to be in front of you today. So awesome. I'm very excited. So before I start, I want to ask a question that David had in his, can you still hear me okay? Okay, try to get that feedback from happening. But I want to ask a question that David had in his advertisement around my talk, because I knew that I wanted to really dig into this and talk to you about it. And that question is, how many of you now or at some time in your past have thought that you are a product of your education? Awesome. That's awesome. Thank you for the participation. Me too. I had that perception as well. I don't have that perception any longer, and I hope that I can give you some sense of why I changed that perception. A product is something that's manufactured, packaged, and for sale. So think about that. And then from there, I'd like to just kind of switch over to say that the way that I was thinking about my future when I was in college, maybe a little bit before I chose my major when I was still in high school, is I thought about the decisions that I made were going to be something where I needed to click into an assembly line. And maybe that's because I was thinking about pre-med, pre-vet, and then boom, 8 years, 12 years later, out I popped as a product with, what happened? That's what I imagined for myself. And I was a little worried about that. And I wanted to kind of gain some perspective around how that might be different. And it took me probably 10 years to really figure that out. Instead, I would postulate we aren't products. So this is what I'm going to call my Frogs on a Lilypad presentation. And I did make it exactly just for you from scratch, but it's been banging around in my head for a while because I use this analogy of frogs on a lily pad a lot, because I think of myself as having a perspective on a lily pad with adjacent lily pads, and I can pick any one of those. I can hop or I can even swim if I really have the courage to take that risk, right? Any new lily pad I land on, with every hop perspectives, and we have new opportunities. And I think that perspective, that mindset, has driven what David would call my non-traditional path. So the title of my talk just juxtaposes STEM education with business. So this is a culture steeped in science education versus a culture steeped in capitalism, essentially. These are two sides of a character from Breaking Bad, for those of you who have seen the series, two sides of Walter White. So the amazing thing about this show is how different these two sides are. And I would say if you could shed the perception of the product, that we are a product, these two are much closer together than you might previously have thought. And so that's what I'm going to focus on today. Here's the pictures are just for your entertainment. People tell me all the time I look nothing like my old pictures. So here's sort of an illustration of my path, and I'll try and move through this quickly because I'm really focusing on the transition point. So I graduated undergraduate with a pre-med degree, decided I wanted to hop on a different lily pad and assess what was around me at the time. And so I went into teaching, and I taught for 5 years and loved it, really enjoyed it. Contributed to a science framework. It was really very stimulating, but I knew I was always going back to graduate school. So I went back to do my master's, and that led me to doing my PhD. And then the big transition halfway through my career, essentially, was moving into a company called Illumina. At that time, it was a mid-sized startup that wasn't profitable. 200 people. It's 7,400 people now. I left there last year. But that decision not to do a postdoc was grounded in my curiosity around more of a breadth of what genetics could do instead of a depth of what genetics could do within the marine science world. So my PhD was in marine science. And then about halfway through my time at Illumina, I spent 14 years there, I made a decision to go from an R&D project management role into a commercial role, which means essentially sales and marketing. So that's the STEM into business structure. And when I look back at these pivotal transitions, some of them milestones, some of them just decisions I made, I tried to characterize for you, for this presentation, what did it take for me to make the right decisions for myself? Because these, I'm convinced, were the right decisions. I have no regrets. And so I'm going to structure my talk around exactly that. I came up with all the C words. So I find that I make the best decisions for myself when I'm calm, when I can tap into my creativity, when I'm connected to people that I work with and trust. And so I decided I would use these words, or a few of these words, in order to structure the stories that I'll share with you today. A little bit of science, a lot of my path. And I just so appreciate you being interested in being here. So specifically, I'll focus on curiosity, connection, and courage, because I think these are the three most important for making the decisions that I made. And I feel like when I can stick with these traits, when I can be curious, connected, and courageous, I make the best decisions for myself, and life really delivers for me. So here we go. We're starting with curiosity. So people use the term learner's mindset for curiosity. And I would say curiosity is the #1 reason I went from that postdoc, I steered away from that postdoc after my PhD toward the biotech, because of the breadth and the genetics and the fascination. I was just fascinated with the technology. The reason, well, essentially, so I went into Illumina, and I will talk a lot about Illumina, but next I'll just essentially tell you some of the things that I learned there. So some of the reasons I went there were that I was amazed by the technology, and then essentially some of this that I think might be interesting to you about just the genetics of humans in general, and the amazing pace that we've seen of discovery in the human genetics realm that is fueling our personalized medicine approach, or an attempt to move into personalized medicine, more than we have in the past. So I went to Illumina in 2004. The Human Genome Project was a federally funded project to the tune of about $3 billion. There was a completed genome, which just means that we now had some sense of the structure around the chromosomes of a human genome for the first time. We knew, very little until we actually got the price of sequencing low enough to really characterize the human genome. And what that means is the first individuals that were sequenced along these milestones of price. So in 2006, it cost $20,000, so that was a significant drop. In 2007, $2 million. We're getting a little closer, but that's still too much to really characterize diversity in humans. But through intellectual property that really hit around here in 2008, and some of that intellectual property was with Illumina, was acquired through an acquisition with Illumina. We were able to then eventually in 2014 get down to about $1,000. Once we're paying $1,000 for a genome, for sequencing a genome, now we can actually start filling databases and start understanding diversity with different populations. What's the difference in diversity between Northern Europeans and Southern Europeans? especially what's the diversity within Africa. We had very little information about diversity in multi-ethnic populations. And so I'm going to talk a little bit about a project that I was fortunate to work on, where Illumina was enabling 2 federally funded consortia. One was called the PAGE Consortium, Genetics and Epidemiology, so essentially looking at health, trying to connect genetics with health, and the CAPA Consortium. Should I pull it further away from myself, or? Okay. CAPA Consortium was essentially driving an understanding of African American ancestry for the purposes particularly of understanding inherited risk for asthma. So these 2 projects were being led, and they were already consortia, what we would call consortia, because they were driven to build groups to do this work by the funding bodies. So Illumina worked with them, saw the different diversity of populations that they were targeting within these consortia, and here's just a list of those. You can see that PAGE was quite complementary with CAPA, and public data really gave us a good characterization of the European Caucasian population, which includes, North American populations, a lot of North American data as well. So given all of this information, there was an opportunity for Illumina, if they could help this consortium, these consortium members, Illumina could accelerate the pace of discovery through access to sequencing, through accelerating their filling up these databases. And in exchange, the thought leaders who were driving these consortia would work with Illumina to help develop testing tools to fuel the next generation of chips, array chips, what we call genotyping chips, that would help us connect disease with areas of the genome. These are called associations. And next I'm going to show you a 20-second video. This is essentially images that were taken, or that were created 4 times a year from 2006 through 2018. Those fixed images illustrate all of the associations, all of the links between the areas of the genome to genetic disease or traits of interest that were discovered from that time historically. This will give you a sense of the pace of discovery, and what I want to illustrate here is how that pace shifts in 2014 once reagents for driving DNA sequencing became much more affordable. These are 23 chromosomes. Next to the chromosomes are dots representing diseases of interest. And they're mapped to regions of the genome. So based upon studies, so at this moment in time in 2015, there were 2,903 studies. All the dots represent the associations from those studies that link disease or traits to areas of the genome. And from 2014 forward, essentially, oops, we might have to play that again. Yeah, now that you know there are 23 chromosomes, right? And this is available, on the website, on the public website. So I'll stop it at 2014, and I just want to show you lots of discoveries made up to that point. Roughly 20,000 associations were known at that point. Now we gained a massive understanding of the true diversity of humans. And the number of associations essentially more than tripled. So the pace of discovery is what has enabled things like 23andMe, Ancestry.com, these tools that are direct to consumers, but also it's enabling our ability to know what to look at within our genetics in order to know our predisposition for disease. Genetics has a small influence in a lot of disease, but in a few diseases, it can have a very large influence. So this is, again, something that I learned during my time at Illumina, and I had come from a marine biology background, right? So working with these thought leaders was, it was just like euphoria. I had so much fun. And I made connections because I was listening to them. I was asking questions. They were respecting my curiosity. And those connections are those types of connections are what I want to talk about next. So in talking about the Lilypad analogy, bringing us back, curiosity tells me which Lilypad I'm going to jump on. But connection increases the number of Lilypads that I see at any given time. The more connections I make, like with David, with Mary Lynn, with those of you that I've met here today and will talk to after the talk, all of those are so valuable for opportunity in our lives. My next story, I'm going to, going back to the story about the Kappa and the Page consortia, what came out of those product development collaborations, essentially, were arrays like what's called the multi-ethnic genotyping array, the mega array that Illumina still sells today, that is amazing for doing those associations, for doing studies of people with a disease, people without a disease, and you genotype all of them on this tool, and that's how you can narrow down what area of the genome might be implicated in any given disease. So here I'm showing a purple line illustrating an area of the genome. The tick marks are one of 2 million variants that are being queried that were fueled by the discoveries in those consortia. These 1 to 2 million marker tools are expensive. The next story I'm going to tell you about is about discovery and development of a tool in agriculture that was, the first tool we built was $30 million. Illumina had no idea existed out there. It was identified, it was driven, and it came to fruition because of building connection through curiosity and understanding what was needed in the agriculture world in order to really make production in agriculture more efficient. So this is a fun story for me. It's not in the human world, but it's one of my favorite experiences. Since a 1 to 2 million variant array is too expensive for most agriculture crops or livestock species, we ended up making a 50,000 array, 50,000 variant array. And this was the very first one we made was specific to the bovine dairy industry. So it turns out that when you have 200 years of phenotypic records, meaning 200 years of trait information around favorable traits for milking in the dairy industry, so these are creaminess of the milk, these are lots of females at birth, those are favorable traits in the dairy industry, volume of milk, young age at first milking, these are all traits that they have collected data on for 200 years. With all of those data, with a very narrow diversity, so unlike human populations, Holstein dairy cattle are much less diverse. And also given an environment where we can select the candidates for breeding, we don't really do that in human much, a variant tool. Sorry. Thanks. And so we worked with the bovine dairy industry to understand what content was necessary to do this, and they taught us how it would be deployed. And I just want to share that with you because this has transformed the dairy industry. If you take a reference population where you know what I said, like I said, the phenotypic traits, the physical traits around milking that you care about, if you take that reference population and you genotype it, now you have an ability to see all the little chunks of the genome that are tagged by those variants that correlate with favorable traits. And instead of actually doing a publication on them and saying, this is milkiness, or this is creaminess of the milk, this is volume, instead you just know that it's a conglomeration of all those favorable, what we call favorable traits, and we can just assign a score to it. So in In agriculture, in livestock genetics, we can then, with a rubric, with a scoring structure, which we call genomic estimated breeding values, we can then just take the offspring when they're born and assign an expected value for them, for retaining them in the dairy industry. And this is enormously successful. So each of those offspring gets a score, you create a threshold, select the candidates for breeding that are above that threshold, and move on. And then you test and you see how well you did. More than 20% improvement in value of the dairy industry in the US. That's been expanded globally. So incredible increases in production. And it turns out Illumina received awards for actually just being involved in that. So it was a pretty exciting time. After building the first 50K array, remember 50,000 variants, Illumina worked with that same community and built a high density array, a lower density array for different purposes, some for beef cattle, some of it was for other breeds within the bovine industry of beef and dairy. And then even more specific tools were built by partners that were specific to genomics and agriculture. Here is a screenshot of the title of one of the publications that came out of this work. After we worked with this group, and we extended this consortium beyond that original group, so we sort of had our steering committee, and Illumina sold, $30 million worth of arrays globally. That group was so happy they worked with us that they recommended us to the pig breeding industry. And they recommended us to the sheep breeding industry. It turns out with the high value animals like livestock, it's much easier to get that return on investment, right? Because the price of food is going to limit how much can be afforded to spend on genetic testing. But so we received an award for the bovine work. We received an award for the porcine work, the pig work. But then we built these tools through discovery with so many different communities. So now I'm just going to march through some of these. Soybean, that was a big seller. Grapevine, maize, pine, pea, wheat. We made several iterations of wheat chips as we learned more about the wheat genome, really complicated genome. Forest trees, eucalyptus specifically, chicken, eggplant, rice. We made lots of rice chips, peach, apple. Each one of these I looked at as a species-specific submarket within agriculture that required slightly different approaches to ensure that the test was low enough in cost that it could provide a return on investment, but wasn't outside of what would be sustainable for those markets. These were built at a time when genetic modification, or GMOs, were receiving a really bad rap with the public sentiment. And so the seed companies were really keen to funnel more resources into accelerating traditional breeding methods, which is what these tools would allow, over those transformation objectives. And not to forget the species we all have in common. These are some of the publications around some of the chips that we built that were specific for looking at specific diseases, or in this case, the African-specific genetic variation that was informing asthma risk, in particularly in African Americans, which is what the Kappa Consortium was funded for. We made several iterations of chips that were specific for cardiovascular and metabolic disorders. So looking at those areas of the genome that had those dots that were specific to some of those disorders. Here's another one that is near and dear to my heart, was building a neurodegenerative disorders chip to identify disease risk around regions of the genome that are specific around neurodegenerative diseases like Alzheimer's and Parkinson's. versus psychiatric disorders like depression, bipolar disorder. So that's my section on connection, but you can see that connection's not in a vacuum. Connection happens, and through connection, you become even more curious. Through curiosity, you gain new connections, right? But now you have the Lilypad you want to go to. You have lots of options for Lilypads, but what you need now is the courage to actually jump. So next I'm talking about courage. Just one story here. When I was at the National Marine Fisheries Service, which is a division of NOAA, I worked on fish. I looked at marine protected areas, used genetics as tools, tried to understand the scale at which we could make effective marine protected areas based upon how the genetics flow of these animals was moving in and out of those areas. I worked very closely with a couple of friends who worked in the dolphin genetics realm. So they were characterizing dolphin communities and understanding the scale at which we needed to conserve dolphins, because it turns out if there's more populations of dolphins, we need to put stricter requirements in to prevent their unintended deaths. So Karina Milmer and I, good friends, I went on to Illumina. We hired Karine 6 months later. She's still there. She and I and Milmer were troubled for many years after Illumina purchased the sequencing technology by the lack of access to these latest cutting edge tools that the conservation community had. They just weren't able to afford sequencing or genotyping or the array technology. So we decided we were going to do a grassroots effort within Illumina in order to drive a dolphin genome assembly. The assembly for dolphin was atrocious. They had no structure of chromosomes. They have a few sequences in the public domain. But we thought if we really understood the structure of the dolphin genome, then we could hang off of that all of the conservation information and characterize diversity within, or start to characterize diversity within dolphin. So we wanted to build this assembly, publish it, and ensure that it was used for conservation. It took us years to get this done because it took us time to get all the right pieces in place. So remember I said I wanted conservation to have access to technology. But that's not all it took. We actually needed a sample source. We went to the NMFS, National Marine Fisheries Service. Those samples were too old. They just didn't give good data, not for doing an assembly. So we went to SeaWorld and we got a live sample from Sandy, a bottlenose dolphin that's still alive there. She's had offspring there. We worked with something called the Ocean Discovery Institute, which is a STEM education group in San Diego. I'm on their scientific advisory board. And we found a student who was just graduated from high school. She was just starting university. We taught her how to do the library prep. She helped us collect the data for the project. She now works at Illumina. She's been working there for several years now. I consider her a huge success story from this project. And then we recruited people from connections that I had made through building all of those consortia at Johns Hopkins and at a company in Israel called NRGene. And then we made sure that as soon as we pushed the button on the publication of it, that those data would be used by the Navy Foundation, National Marine Fisheries Service, and the SeaWorld partners to actually drive impact. So we were super excited to get this done. It turned out to have a lot of visibility within the press. Remember, we were doing this grassroots, like Illumina kind of said, oh yeah, well, if it just makes you happy, go do it. We recruited a dozen Illumina employees that donated their time to do it. And when it hit the news, Illumina was thrilled. They were so visible, and everyone loves dolphins. So we were able to then look back and say, what was it that made it successful? That's when we identified the 5 components. Now we've created what we call an iConserve kit. And just 3 weeks ago, we got approval to replicate this conservation concept across 20 global sites where Illumina does business. So it feels like we're actually going to make an impact that's much more broad than we would have made if we had just helped a researcher do a dolphin genome assembly. Instead, we built it from the ground up, and now we have something to replicate. The reason I tell you this is essentially, David, you might kill me for saying this, but you don't always have to ask permission. Sometimes you push it if it's the right time. I mean, we wouldn't have done this if Illumina wasn't profitable, right? If it's the right time, it's the right thing. You've got local support. Sometimes it gets so big that it becomes official. And that's why I think this is a reasonable example for courage. The other topic I'm just going to touch on, and then I'm going to wrap it up, is the courage of self-awareness or self-reflection, which I think takes the most courage of all, honestly. And so something that has helped me in this effort is having some structure around identifying what works for me and what doesn't work for me. Recently, I found a book that gives a nice structure that I want to share with you because it makes it more accessible to you. This is from a book called Designing Your Life. It comes out of the IDEO lab at Stanford, their design school. Human-centered design, recommend it if you want to research it. I recommend them. I recommend Luma and resources for learning. This is to build an activity log for 3 days of all of the things that you do in your job, at home. For every activity, score it as to how engaged you were. Like a maximum engagement is when you go into flow. We all know what that feels like, where time just passes and we don't even remember, where we're at. That happens when my husband comes in and says, "It's time for bed, honey." So knowing what times and what you're doing when you go into flow, super powerful. And then understanding what gives you energy versus what depletes you. So I've found this enormously useful. I do it every quarter, 4 times a year. I characterize what I'm doing, what my activities are, and what is working for me. And the book actually gives you some more structure around digging into the reasons why anything might be giving you that charge. But to maximize your productivity, but in a way that is incredibly satisfying to you, just feels like, why wouldn't you do more of that? So that's the end of my section on structure, or sorry, on courage. Back to, are you a product of your environment, of your school, of your education, of your decisions? So products, they tend to be launched to capture a specific market share within a larger organization. They have clear fixed requirements for manufacturing. In order to rev them, they require very clear steps and guidelines around quality assurance and quality control. I would argue maybe you're more like a spin-out. And you're a spin-out, sure, of your education program, of your past jobs, of your family. But as a spin-out, and spin-outs are usually spun out of a parent company. Illumina has spun out several companies, one on forensic genomics. A spin-out is typically launched to address an unmet need, but to allow the parent company to focus more, right? So it doesn't distribute the resources within the parent company, but it's still valuable. And so you spin it out in order to meet not just a market share, but a real unmet need. These are entrepreneurs, right? Usually it's maintained with an interest to the parent company. So there's your connection. And you have to pivot. You have to be agile. You have to fail fast and move on in order to stay in alignment with market demands. And so I like this idea much more than I like the idea of being a product. And finally, my last, have a conversation with you. So stay curious, foster your connections, Everyone should have an advisory board. I have one. The people you go to who have a vested interest in you and you have a vested interest in them. Muster your courage and be more than a product. Thanks.
Thank you, Cindy. Hey, questions? I'm in a row with mics. Does anybody have a question? Don't be shy. Come on.
Matt?
Yeah, hi, Cindy. I thought that was great. I'm curious, what would be one...
Curious, you're curious. I love
Yes,
it.
already curious. What would be one actionable step that you would recommend for any student who wants to sort of enhance their own personal development?
Read. You know, Warren Buffett, I believe 300 pages a day, does he say? 500. Oh, I love that you knew that. That's awesome, Proteus. At least 2 hours a day. So he reads books, or he talks about reading books, but it doesn't have to be books, right? It can be scientific papers. It can be information on the internet, right? Just know that sometimes they're just advertisements. So I think it's reading, right? Reading along with connection builds your network. And then reaching out to people that have written something that you're really interested in, boy, they on my door. Most of the books in Barnes & Noble sell less than 1,000 copies. They want to talk to you. Thanks for asking the question.
I have a question about your selective breeding. If there was any unintended consequences, and what I'm thinking about is in the dog world with the many breeds of dogs, some die early, some have bad hips, some you get asthma and all that kind of stuff.
Right. Oh, I love the dog analogy. I have so many opinions about dog genetics. And I'm gonna stick to that one. So the unintended consequences that happen have to do with not knowing when there's some hidden recessive trait that then gets amplified because the animal has so many great favorable traits. I'm thinking of bovine now. So there are great examples we know now of historical mistakes we made where I can't remember the trait off the top of my head, where something was perpetuated through an entire line and they can actually attach how much value was lost in the industry by having that trait remain. If we had known the genetics, we would have been able to avoid that. With dog breeding, I think what's needed, we're not going to stop having AKC breeds, obviously. There's so much passion around it. But what I think is needed within there, and I think we're moving in this direction, is that there needs to be a website for every breed. Within that website, all of the registered animals that have been genetically tested. You then, if you're going to breed these animals, you then have your animal in there and you select the animals that are available for crossing your animal. And if the diversity gets improved of the entire breed using a score, then that's an okay selection. If the diversity gets squashed by doing that cross, then I would throw up a flag. And then beyond that, there's all the fixed traits that these animals might have that you want to avoid. I don't think it's the right thing to do to take an animal out of breeding just because they have some genetic issue. It depends on the genetic issue. But like one of my girlfriends has an Australian Shepherd, it has an extra rib. I wouldn't take that animal out of the breeding pool, because every time you take an animal out, you reduce diversity of the breed as a whole. So as much as possible, we want to keep those as diverse as possible. Does that answer your question?
Well, I was thinking about some of the other things that might have come up as surprise. You started talking about having bovine. Is there anything else that maybe the pigs or chickens or anything else?
I believe it's, this is my opinion, I believe that the ability to avoid that is enabled by having some visibility to the genetics. So I think that's less so now than in the past. But there's always unintended consequences when you're inbreeding. right? So for those of you who don't know why agriculture does all of these monocultures, you think about growing a wheat crop. We all have thought, I've learned in my genetics training, diversity is better. The more diversity, the better. But if you plant a bunch of wild seed out in a field, you're going to have some of it coming up now, some of it coming up in 2 weeks, others coming up in 3 weeks. And the costs of actually harvesting become astronomical. So the reason we want favorable traits stacked into a particular seed type, and then we want to have that same seed duplicated for the entire, or replicated for the entire field, is because we want it to grow up at an even rate so that we can afford to actually harvest. So it took me a while to learn that. I didn't know it until I started talking to people that were enabling those decisions around breeding. Christine.
Thanks so much. That was a great talk. So I noticed when you came through pre-med, but you did not go to the medical school, which was very brave / courageous. And all these students are going into bioengineering because there's such an easy pathway. What is the new path? What is the courageous path of not bioengineering, but the next one that's not? What would you say that would be?
You know, the big areas now are being able to handle large data sets, but being able to handle them not just in, you know, an environment of Google where there's just enormous amounts of data about people, but being able to do bioinformatics with those large data sets. So being able to manage those data from biology. So when I was in school, we didn't have bioinformatics. It's a field, you should all look at it. We had computer science people, and then we had biologists who liked to be at their computer and not talking to people. Those are the two things we had. So, I mean, computer science, you know, you could argue some of them have self-selected because they get to build things, right, and be at their computer all day. So we had those two choices. The computer scientists didn't have the biology language. It's a vocabulary, and it takes a lot of memorization. It takes years to memorize all those words. Hi, Alex. I didn't know you were here. So we had computer science people that were trying to teach biology, which was challenging, especially if it wasn't their passion. And then we had biologists that we were trying to teach computer skills to. Which one of those do you think was more successful in doing the bioinformatics before we actually created degrees in bioinformatics? Computer science, how many people? Biologists? Yeah, absolutely. Never underestimate the power of vocabulary. It's amazing. So now we have that field of bioinformatics, right? So at any given time, sit down with your scientific advisory board, you know, your mentors, and start brainstorming around what are the missing pieces in careers that we haven't created yet at universities like that, right? And see if any of them appeal. And then I really think it's valuable to be cross-functional. And I think that we can't undervalue the soft skills, or we can't overvalue the soft skills. The soft skills like courage, like curiosity, like connection, right? But reading is still really important, and knowing the information so that you can know what excites you. Yes, Bonnie.
Hi, Cindy. So you started to kind of go down the road that I was going to ask.
Bonnie and I did our graduate work together at Scripps Institution of Oceanography. She's a professor at UW Tacoma.
So I'm actually asking a question from the UW Tacoma perspective. So given that when you and I were in school, there was no such thing as bioinformatics, but there is now. As we're trying to plan education for our students, we often think it's like we're trying to teach our students to have the ability to answer unscripted questions that like don't exist yet.
Like the one you're asking now.
Well, yeah, I'm formulating it as we're talking. So thinking about how do we prepare students, and you started to go into some of these softer skills, how do we think about designing curriculum to make the students of today flexible as you've been sort of going through your career? What are some of the things you might recommend to us as educators?
I would bring in people that have a different experience, people that haven't done traditional paths. I think David's got it right. I think, you know, I don't think about my path as non-traditional or traditional. I just think of it as my path. But David did, and I was like, oh, there's value in that. Okay, let's do the frogs and Lilypad talk. It's finally my opportunity to finally put that talk together. So yeah, I think role models. It's weird to say that because now I just called myself a role model. Yeah. Did you have a yeah?
So you started out in the early part of your talk. You basically mentioned how a certain process went from being very slow and expensive. Shall I? In the early part of your talk, you mentioned a process that was originally slow and expensive and got faster and cheaper.
Right.
And then in the middle part of your talk, to my ear, basically the agriculture story was kind of a developing crystal balls kind of story. Like how could we use the genome to look and see things about the phenotype of the eventual phenotype of these animals that we can only be seen if you have the crystal ball. Did you learn anything, or could you share any insights about developing crystal balls more efficiently from that experience that might be a valuable lesson for what's happening here at RAIN, just in terms of, I don't know, tapping into or...
Well, you just keep layering on that question, don't you?
Yeah.
One after,
Because
I had it, and then you... No, I'm just kidding.
Take any piece of that and riff.
So before you said to make it applicable at RAIN, although I think, I know what RAIN is doing now. I think there are boundless opportunities for what RAIN can do in the future, but being able to build predictive models is what personalized medicine is all about. In particular, in immuno-oncology. Right. So let's just say oncology. So in cancer, we used to just try and kill all the fast growing cells that were in your body and hope for the best. And that was chemotherapy because that was the best we had. And it actually worked really well in a lot of cancers. Now what we're trying to do is we're trying to characterize the cancer. And it turns out that tumors are very heterogeneous. They're not simple. We have to take lots of cells from there and to understand what's going on there. We need to characterize what's happening in those tumors to know how to turn on our own immune system to actually attack them. Some cancers seem to do that well, and crazily enough, the microbiome seems to have an influence on whether people are responders to certain therapies, cancer therapies, and versus non-responders. Like, it's such a story. Come talk to me afterwards if you want to hear that. It's so interesting. But essentially, we want to understand what's happening in that tumor. Are there immune cells infiltrating that tumor? Are they replicating? Are they part of the problem? Are they part of the cancer, right? So a lot of times cancer evades the immune system because it's self. It looks like self. How do we turn it on so that the immune system can see it? This is as predictive as I can imagine ever being. Like, how amazing is that? It's an amazing area of study. And these days, doctors that come out of medical school have options in research that they never had when I was in school. And I didn't know where it was all going to go. So it's not, like I said, it's not clicking into an assembly line anymore, even with an 8-year path to medical school. It's very different. It's fun. Yeah, Stan.
So I thought it was fascinating when you brought up the journal about engagement and energy.
Designing your life. I just love the designing your life.
It's more about, so you talked in the beginning about how we treat our agriculture and our livestock to try to hone it to be the best it can be. We also trying to do this with our own genetics, right? But at the end of the day, it's that component that we don't necessarily pay attention to. So I guess
which component?
The component of yourself, of your energy and your ability to focus and give your time and into something that creates and something that drives creativity or drives accomplishments or products or whatever you want to, whatever the work of your labor is. did you find by doing that engagement and energy for your task, did you find that over time you were able to actually find more engaging and energy saving tasks that filled your list,
Filled my cup.
filled
Right.
your cup? Or were you able to
Yeah.
actually maximize that like you would, cloning a genome or, selecting for certain traits?
Yeah. Every job has a mucking of the stalls, right? Everything we do will have some stuff that's energy draining and not ideal. Every job. And I'd argue that's okay because our minds need a break sometimes. Sometimes I want to listen to that audio book while I'm combing through data. But I would say definitely I have more of what engages me than I ever have before. So it works for me.
I have a question.
I have no doubt, Proteus.
So let's say there was a hypothetical young entrepreneur who,
All right, let me work on that. Okay, I'm ready.
let's say there was, who was interested in an area of science and technology that was controversial yet mind-bogglingly important. How would that entrepreneur encourage others to be curious about this area? And how would that entrepreneur connect to others who could form a team that would work on this important area of science? And how would that entrepreneur have the courage to not ask permission, to pursue this area of science in spite of the opposition, and boldly go where no one has gone before?
I love it. He's awesome. So I'm gonna start with your initial, finding people that can resonate with your message, with your beautiful business proposal, which I find amazing, and which will refine over time. Like we've talked about, there's no substitute for talking to people, but do it in a way that collects pretty objective information. So almost a survey, right? Where you're asking the same questions in the same way to understand which aspects of your plan are setting off alarm bells, and how do you mediate that, right? How do you adjust your plan or shift your plan so that it's going to be more palatable, right? So we've talked about the area of genetics where we are editing livestock. That's a pretty sensitive topic. People are a little worried about gene editing, right? So the group that cares about driving this innovation and have created a startup around it are focusing on animal welfare traits. So it turns out that bovine are born with horns or not with horns, without horns, not based on whether they're male or female, which a lot of us thought when we were in elementary school, right? I drew all my bulls with horns. It's actually genetic. So some cows have horns, some cows do not. It's a gene. That gene was identified because of the building of those arrays that allowed them to find the area in the genome that coded for that. Now they can query and know when an animal is born if they're going to grow horns or not grow horns. If they grow horns, it's necessary to remove them if they're going to be kept in a dairy livestock environment because of the safety of the other animals. If you can edit to have all animals have no horns, you've just removed the necessity for a very expensive procedure that is not fun for the cows. So focusing on areas that can, to some extent, you want to be brave and push outside, but you also want to be successful. So there's a balance, right? I love the global warming thing myself, just saying, yeah. You guys should talk to him you should be part of his survey. Yes, Matt.
You talked about the impact of accessibility to technology and specifically genome technology. What community would you like to see get more access to genoming technology?
I'm sorry, conservation. National Marine Fisheries Service, right? The burden of proof that there's a problem with our fisheries is on the fisheries managers. They have to prove that there's a problem before we can put fisheries regulations in place. That bothers me. They came about, we put that in place because we looked out at the ocean and it looks vast. It looks like there's so much out there. We'll never run low. But it turns out there's actually a pretty small ecosystem there where these fish are reproducing. And so we can overfish them pretty easily. So I do have a very strong tie to that. I care about that because for 20 years I've seen that it's really hard to have a conversation around saving the planet without just having people roll their eyes. It's not a sexy topic. It interrupts progress. And so trying to enable research that will help us make scientific decisions around that will be much more acceptable and easier to sell. It's a business proposal, right? We need to save these fish so our kids can eat fish. One more here. Oh, whoops.
Hi there.
Hey.
Jen Greenius with Resolve Conservation. Thank you for your talk.
Oh, yay, conservation.
Right. So one thing that you can't go here into the Puget Sound area and talk about conservation without talking about orcas particularly. And so I was really interested in the dolphin case study with Illumina and how you see perhaps lessons learned or best practices that could be potentially applied to our orca problem here.
Sure.
And in terms of the dolphin, when you in that cycle, what were some of the findings that changed the policy and the actions for Navy Foundation and all of these groups? Like what did you actually glean from that?
So we just published it end of last year. So it's just come out, right? And so researchers are using it. You can think about a genome assembly, kind of like the Human Genome Project. It's sort of like the bar in the top of your closet. Without that bar, your clothes would just be a pile on the floor, right? And trying to figure out what you want to wear would be very difficult, right? So being able to have that structure in order to make comparisons of using the genetic data we do have, and in fact, we actually have a lot of genetic data on populations of dolphins. We just didn't have a great context in which to interpret it. And so what we believe about the assembly is that it provides that context to better understand the data we have. Does that make sense? So I would say we need assemblies for certainly every whale species needs a genome assembly. And so I would say, is that really going to move you in a direction for improving decisions around the stock? Or is there a better question that we should answer? Should we instead do deep sequencing of 150 animals to really characterize how they're inbreeding or how they're responding to their numbers? I don't even know how many orcas there are left. But so I would say, what is the need? What's the need? It's always what's the need? What is helpful? We picked the assembly because we could do it from within Illumina. It would be impactful. And the marine mammal world, people don't talk to each other very well. And so it was hard for us people would not share what they were doing, even with the prospect of getting some sequencing done. And so we just did what we thought was needed. Now we're in more of a position, or certainly Karine, who's still at Illumina, is in more of a position to have the conversations to understand what's needed. And Illumina's technology has advanced to the point where I think this sort of doing population level studies would be very useful. I mean, ultimately in conservation, you want enough information. Let's think about animals like wild dogs in Africa, which are also endangered. Once you move them away from, cities, do they ever reproduce again? Are they interacting with other animals? How is the leader of the pack structured? Is he responsible for all of the offspring? Understanding the population dynamics can be informed by just taking poop samples, sequencing them, and comparing them. And so that sort of thing is boots on the ground work that needs to be done and needs access to this sort of technology. I think you had a question.
I did. So, and you didn't go there in your presentation. So if you don't want to answer this question,
I'm really brave today.
Well, so bravery. If I read your first slide right on your history, you're not at Illumina anymore. And your whole,
Yeah.
you're pretty passionate about what you've done there and the effects.
14 years,
I mean,
right?
it's pretty cool stuff.
Yeah.
What kind of curiosity? I just want to hear what led you to leap off that lily pad, and if you'll talk about what you're doing now.
Sure, sure. So I leapt off that lily pad. You saw the picture of me with the dog at the beginning, right? So I leapt off that lily pad. I cut my hair. I took 6 months off and got a puppy, which gave me a lot of perspective, and it was a lot of fun. And then in November, I started consulting. So I actually, several of my former customers, because of those connections, were reaching out to me to ask if I could do work for them. And so I've been doing a smattering of different things based upon my skill set to stay connected with those people, to pay the bills, to give me time to decide what my next lily pad is. So it's kind of like what I did with teaching. I taught high school. I thought I'd do it for maybe 2 years. I thought these are good skill sets. My mom was a teacher. You get to manage high school kids. Like, what a good skill set that is. And I stayed for 5 years because it was so much fun. And I built a marine science program in a school in Central California. And eventually it was time for me to go back to graduate school, and I knew what I wanted to do. And so there's a lot of trust too in just knowing that it'll be right when you see it. Thanks for your interest.
Okay, let's give Cindy a round of applause. Thank you so much, Cindy. So thank you guys for all coming tonight. If you have a minute, you should go up and talk to Cindy, engage her, but also just recognize the amount of talent that's in the room tonight. I know a lot of you, and there's some incredible talent here in the room. Taking a minute just to talk to somebody and network, you easily realize how we could create new companies, how we have opportunities for education for all of our kids in this community. And so it is extraordinary what I see here tonight. So especially the women representation in the room. I mean, I know some of you guys here, so it is absolutely amazing. So, and again, these are role models. These are role models for our kids. You know, this is a community that I really think is ready to explode. And so I will continue bringing talent in from outside, but I also want you to recognize the talent that's here too. So let me know, help me continue to do this. Let Cindy know how appreciative. Mary Lynn also came up here from the South, Southern California. So, oh yeah, sorry, Southern California, but yeah, San Diego's, it's south of here, you know, little town south of here. But recognize that, you know, we can pull the talent up, but I'm basically showing you examples here, but we have that here. And so that's really what I want this community to realize, that we can do this. We can do this. The technology that we have is, we're emerging this in the lab right here. The research problems we have in our community are here. We have the marine environment, we have the medical environment, we have the joint base right here. So you just need to ask questions and come ready to do some work here. So thank you again for coming tonight. Talk to Cindy, talk amongst yourselves.